Hello, everyone. My name is Kathleen Morrill. As Diane mentioned, I'm a member of Eleanor Carlson's group here at the Broad and also a PhD candidate at the University of Massachusetts Medical School. My dissertation work is aimed at understanding the relationship between genetic variation and behavioral variation in one of the most iconic and diverse species we know, the domestic dog. Much of the data I'll be talking about comes from our public science initiative, Darwin's Ark, which collects behavioral surveys and genetic samples from thousands of pet dogs. And what I'll be sharing with you today is the story of dog breeds and how these, frankly, very weird subpopulations of dogs might help us explain the genetics of behavior and personality. But more importantly, I'll describe how we can understand these breeds from a population genetics perspective. I'm going to kick it off with a few definitions for PopGen processes at play in any population, including dogs. The first is genetic selection. Genetic selection is the non-random process through which phenotypes influence the changes in allele frequencies across a population. When someone says natural selection or artificial selection, it's really an attribution of the cause of selection, so such as from survival and fitness in the wild versus human-directed breeding of a domestic species. Genetic drift, on the other hand, comprises random changes in allele frequencies over generations in a population that can be really affected by the existing allele frequencies and sampling and bottleneck effects. Genetic differentiation, from either case, selection or drift, is the accumulation of changes in allele frequencies that distinguish a population from others, and that's what we're going to be talking about today. In today's talk, I'll first give background on the domestic dog and why dogs are both a very useful and sometimes very confusing population to study thanks to selective breeding. Second, I'll review our approach to studying genetic differentiation in the dog breed populations and the multiple ways that I attempted to find what traits are under selection in dog breeds. And finally, I'll give kind of a perspective going forward on how breed ancestry and admixture might be useful for addressing directly breed differences in behavior, but only if we can overcome the unique challenges that dog breeds pose to population genetics. So we all know dogs, and throughout history and even today, dogs have undergone many population events and experienced selection in different ways. So domestication from ancestral wolves involved physiological and behavioral adaptations to anthropogenic environments to be near people. These included things like starch metabolism, increased starch metabolism so that they could eat food that we eat, and reduced fear of humans. The global proliferation of dogs as a species followed, and today dogs exist nearly everywhere that we live, continuing on the adaptations that domestication brought. The majority of dogs worldwide live near human settlements, but don't necessarily live in human homes. And often these dogs are called village dogs or street dogs. And in some sense, they are kind of the default dog in the species. Selection for behaviors, especially those useful to humans, such as herding behaviors that help us manage livestock, occurred under more direct human influence. These selected behaviors often rely on disruptions to the typical wild canis hunting sequence, which is a series of motor patterns. But also selection for behavior still happens today. One really clear example of this is breeding programs for working dogs, for dogs that have to excel at tasks like guiding the blind or sniffing out bombs. The creation of modern breeds, or at least the European practice of breeding for consistency, started in the 1800s and continues today. Breeds are isolated populations of dogs that are bred for consistency and conformity. Many breeds may derive from lineages of dogs selected for behavior in the past. And today, kennel clubs prioritize breeding for conformation to a breed standard of physical appearance and for purity of lineage without introgression or outbreeding to dogs of outside the breed. Domestication is really what captures most people's fascination with dogs, and justifiably so. That was an incredible selective history. But the domestic dog is also interesting in the vast trait or phenotypic variation expressed across the modern population. Some of the traits in dogs have very extreme phenotypes, and some have even called this an example of diversifying selection. Domestic dogs have some extreme variation in their physical traits, in their shapes and their sizes. Dogs can also develop health and behavioral disorders similar to afflictions that we too face. And then there are behaviors and personalities or temperaments that differ across dogs and for which humans may have exerted direct or indirect selective pressure. So behaviors like herding that were immensely useful throughout human history, or demeanors that are actively bred for and tracked in working dog programs, or even just suitability as a pet companion or a dog sport champion. In research, dogs prove a unique asset to genetic investigation. Unlike other animal models that have been generated for the purpose of replicating human phenotypes in the laboratory, and whose own phenotypes are considered not really more than readouts or proxies for human disorders, we study dogs in much the same way that we study people. We see dogs as a natural population with its own genetic and phenotypic variation. And we work from the same assumption that there's commonality to be found in, say, the neurological pathways of behavior across animals. And we might learn from the similarities between humans and dogs. But dogs are also just interesting in their own right. Today, it's unimaginable to think about dogs without thinking about dog breeds. These isolated subpopulations in many ways highlight the vast variation in dogs, but they do so by distilling those traits down into these recognizable icons. And although dog populations have existed across the world for millennia, the European practice of breeding dogs for consistency didn't emerge until the 1800s with dog fanciers in Victorian England. And it's all about just showing what is, say, the best specimen of a dog's type. And as we recognize them today, most modern dog breeds, at least in the show world, require that dogs are bred to, firstly, conformation with the breed standard for physical metrics and appearance, and two, purity of lineage without introgression from any dogs outside the breed, although exceptions are made, usually at very high scrutiny of kennel clubs. Not only are dog breeds recognized for their distinctive physical appearances, which are, again, maintained by those breed standards, but breeds are purported to express distinct inheritable behaviors. This belief originates from how lineages of dogs have been selectively bred for different behaviors, temperaments, and jobs in the past, largely by modification on motor patterns observed in the Canis hunting sequence. For example, there are behaviors that help manage livestock or assist on a hunt. We see here examples of eye-stalking behavior in herding breeds, including border collies, behavior that makes, livestock move. Different related herding behavior called heeling, where the dog actually nips at the heels of cattle. And this is reported to be present in breeds like Australian cattle dog and the corgi. Or retrieving, which is, biting prey objects gently and bringing them back without actually devouring them. Incredibly useful if you're on a duck hunt. And the formation of these different dog types for scent hunting, for livestock guarding, for shepherding, probably predates the formation of modern breeds, but you can imagine why breeds might be representative of these past selective histories, and many breeds today are still grouped by their historical purpose, even if they're not actively bred for these behaviors. That said, there's no one story that defines all breeds and what traits are under selection in breeds. Breeds vary in when they were founded, what population they derived from, they vary in origin, and they most certainly vary in selection. Some breeds were just formed as a way to preserve someone's favorite dog. Others were just derived directly from dog populations native to a locale. And others still came about from crossbreeding and just stuck around because people liked them. And as the origins of breeds are difficult to discern from mythology, we need to test our assumptions that breeds differ in behavior and ask what traits are under selection rather than take those stories or claims at face value. So a few different questions motivated my research into the subject of dog breed populations. The first of which I'm actually going to circle back to later, and that is testing that assumption that breeds carry differences in their behavior. And what is the extent of that relationship between breed and behavior? But first I wanted to see just what is different about breeds genetically. What kind of genetic differentiation do these populations have? And from that, can we find what traits are under selection in breeds? Are they physical traits? Are they these ancient behaviors? How how can that be genetic differentiation be useful for finding selection? And then finally, how breeds can be used to investigate the genetics behavior. What asset can these interesting populations serve us when we are looking at their genomics? So what differentiates breeds in their genome? So restricted genetic flow between breeds and their relative isolation and lack of introgression from other dogs has produced subpopulations which certainly have genetic differentiation. Regions of the genome that are most differentiated in breeds may be under selection, or they may have fixed alleles from past selection, or they could just have allele frequencies that are out of whack due to genetic drift. A variety of approaches have been used to identify differentiation signals in populations across the genome. For non-domestic species, these often look at deviations from the frequency spectrum of derived alleles. But for domestic species that may have many recent population events, looking at alleles that are derived from the ancestral wolf might not be the right approach to find regions that are under fairly recent selection. What we will look towards are differences in allele frequency at single nucleotide polymorphisms, or SNPs, in a representative set of dogs from breeds. And what we want to find are the SNPs and subsequently the genomic regions that deviate in a breed relative to other dogs. The fixation index, or FST, is a commonly employed statistic that measures distance between populations and differentiation at genetic variance. FST can kind of be thought of as the proportion of variance in genotypes that can be explained by population structure. And there isn't really one single estimator of FST. There's actually quite a few, and they apply different assumptions, and this can lead to a lot of confusion. So for example, the Cockerham estimator of FST assumes that the populations you're comparing have experienced equal amounts of genetic drift over time, which is almost certainly violated in the dog breeds due to their varied origins. In contrast, the Hudson estimator of FST, which is what we'll apply, doesn't make that assumption, and it's also not as sensitive to differences in the ratio of sample sizes. The population branch statistic is an extension of FST applied 3 ways to 3 populations for the purpose of finding differentiation distinct or more extreme at one population. Its value comes from its ability to mitigate allele frequency differences that come from other or older population split events, such as the split between dogs and wolves. Here, we opt to use all other dogs, purebred, admixed, outbred, as the comparison populations, so that we're studying just the differentiation in one breed relative to dogs overall and not making assumptions about the relationship between breeds, which some breeds are indeed related to one another. Population differentiation has been applied to dog breeds previously, with the primary aim, of course, being finding genomic regions responsible for traits that are under selection in breeds. Firstly, there was a study that calculated the pairwise FST for 21,000 SNPs in 10 breeds. And they found which SNPs had the most deviation in each breed relative to all the other pairwise comparisons. It's this study that identified the gene Has2 in Chinese Shar-Pei dogs, which they further examined for its relationship with an obviously selected trait in Shar-Peis, their skin folding. In a more recent study, the population branch statistics were measured in sled dogs and sled dog type breeds. And researchers found a bunch of differentiated loci, but they were left to speculate what, if any, roles these regions had in traits that might be under selection in sled dog populations. Therefore, genetic differentiation is really the first step, but not the final step, to investigating selection for traits and breeds. It can only reveal what genomic regions are differentiated, not why or for what traits, or if it's just due to drift. The approach we performed was to take each breed, a bunch of publicly available data from those breeds and data we've produced ourselves, and calculate the PBS in sliding windows across the genome. We used 2 populations for comparison here, all other dogs, which includes dogs from breeds, mixed breed dogs, and village dogs, and wolves as the further distant population. The set of 27 million SNPs used have been genotyped in at least 80% of all dogs and 50% of all wolves, such that windows contained enough information to calculate this 3-way FST type statistic. What you have here across the genome, positions on the chromosome, is the mean PBS per window across all 101 breeds. Now, what immediately stands out is that there are some obvious regions of the genome that appear to be differentiated in multiple breeds, the content of which I'll describe later, but it should be unsurprising given that the formation of breeds happened from the existing dog population. Our usage of all dogs as a comparison population, including dogs that are not from breeds and unlikely to be under selection, may be why these loci stand out. Among the top PBS results for breeds, we observe similar patterns to previous studies for traits expressed in breeds. So, for example, the genetic locus associated with blue eye color was originally found in Siberian Huskies. And we also see that it's highly differentiated in that breed. We see this pattern similar for Tibetan Mastiffs, who are known to carry a haplotype that confers high altitude adaptation at EPAS1. And we see high differentiation in that locus in Tibetan Mastiff. So we're seeing things that we would expect to be differentiated breeds based on previous studies. But that locus, ALX4 for blue eye color, is far from the only one differentiated in Siberian Huskies. Like the sled dog study, we find CAC-NA1A. This locus is highly differentiated in Huskies. But across these loci, we can't reasonably assess the content of these differentiation signals or the traits under selection without more information, which leads us to ask, how can we determine which traits are under selection in breeds? Ideally, what we want are a set of genomic loci we know to be associated with different traits in dogs. And we can get these by looking at genome-wide association studies. At their most basic level, association studies compare genotypes across individuals of different phenotypes, and thereby mapping across the genome which genetic variants are associated with trait differences. To date, canine genomics and association studies have focused almost exclusively on breeds, whether by studying variation within a breed or comparing between breeds. The interbreed approach is very powerful because within a breed, there's a reduced overall genetic variation across the genome. And you have more precision with the phenotype in question. So you can be fairly certain that when you're comparing, say, Papillons with a retinal atrophy disorder to healthy Papillons, that is the trait you're comparing between them and not something like their size and their coat color. Similarly with the collies, between the smooth coat and the rough coat, if you're comparing these dogs in an association study, you can be fairly certain that that is the phenotype you're asking about. The interbreed approach often involves using breed average phenotypes and genotypes, again, working off that benefit of reduced genetic variation expressed within the breeds to say that those genotypes are representative of the same dogs from the breed. And these two approaches can both be fairly powerful. The interbreed approach has been used to identify genes responsible for physical trait differences, such as IGF-1, a gene that's associated with differences in body size. This approach has also been applied to behavior, for example, to identify genes that may be involved in canine fear and aggression varying across breeds. That study also happened to identify IGF-1, which really highlights the major confounding problem to this approach. When we compare breeds to breeds, we're really comparing apples to oranges, and it becomes very difficult to divorce the multiple phenotypes that distinguish those breeds, especially between physical appearance and behavior. And often these interbreed approaches don't use individual level genotypes and phenotypes. So a major challenge to performing genome-wide association studies in the pet dog population is obtaining those paired individual phenotypes and genotypes from individual dogs. So we launched Darwin's Ark, a public science initiative in 2015 to address this problem. For over 20,000 dogs, we've collected behavioral surveys and physical trait surveys from validated questionnaires on personality, impulsivity, cognitive decline. We also gather DNA samples and have sequenced over 2,000 dogs at low coverage with imputation of genotypes against a large panel of whole canine genomes. For the first 110 questions in our surveys, which all cover behavior, we performed exploratory factor analysis to identify latent behavioral traits and score dogs according to their survey responses. This approach is similar to a principal component analysis, except that the factors we find don't need to be orthogonal. In fact, there can and will be correlations between factors. The nice thing about these factor scores, though, is that they give quantitative measures to what could be very multifaceted behavioral traits. So these are the first 8 factors we discovered, and they explain 24.3% of variance in survey responses. And we name these factors according to the questions that underlie them. So we have things like sociability with people, sociability with dogs, the context in which a dog will apply aggression, which is our agonistic threshold, toy drive or toy-directed motor patterns, and bitability, which is kind of a dog's amenability to being trained and responding to trained behaviors. So you know how I said earlier that we wanted to test those assumptions that breeds differ in their behavior. Well, it turns out with all that survey data, we're actually able to interrogate that. We tested whether breeds differ from one another in behavior, something other studies have done and indeed found differences. And we do find that behavioral characteristics distinguish breeds from one another, at least in the set of breeds for which we had the largest number of confirmed purebreds shown to the right. We also asked a question that previous studies seldom ask, which is, do behavioral characteristics not only distinguish breeds from one another, but do they distinguish breeds from dogs overall? And namely, we developed a permutation approach that compares random samples of dogs from a breed to a random sampling distribution of pet dogs at large. And what we see is that certain breeds do still come out as significantly different from dogs overall in their behavior. But the relationship between breed and behavior isn't always as strong as we'd expect, and it certainly doesn't extend to all breeds or all behaviors. Nevertheless, we not only collected survey data, but we collected genetic data as well. For every dog's genome, we wanted to get their genotypes so that we could perform genome-wide association studies. In our genetic data set that we use for GWAS, we have 2,155 dogs. A smaller subset of those were genotyped on a high-density chip. Most of our newer sequenced dogs underwent low-coverage sequencing, and both data sets underwent imputation against a larger set of high-coverage genomes. And we performed genome-wide association studies in this diverse pet dog population. Our association studies use a mixed linear model-based approach that accounts for the effects of population structure and kinship or relatedness. Here's shown our genome-wide association study for height differences in dogs. To the right is our distribution of phenotypes from 0 very tiny dogs to 4 very large dogs. On the x-axis are our positions along the chromosomes, and on the y-axis is our association p-value, plotted on negative log scale. Each dot here is a SNP, and as they rise, so too is their association with the trait in question. And you'll be hearing more about the genetics of biology of height and skeletal growth in the MPG talks to follow this primer, so stay tuned. But the blue dogs here indicate that genes in this region have previously been found associated with body size differences in dogs. And the gray icons indicate that those genes have been found associated with height differences in other mammals as well. So this approach seems to be working very well. And so now we have 3 different pieces of evidence or data to consider. First, we have the breeds and the genetics. We have genetic differentiation signals in the form of those windowed population branch statistics across the genomes of purebred dogs. Second, we have breeds and traits. We know what breeds have significant trait differences, and we have the direction and degree to which they differ. And finally, we have traits and genetics. We have association signals for trait phenotypes across a wider pet dog population. So the question is, how do we bring these lines of evidence together and find traits that are under selection in breeds? Well, my first naive approach was to combine differentiation signals for breeds that share traits in common. So we know that both Siberian Huskies and Beagles howl more than other dogs. This is a Siberian Husky, They're very chatty. And this is a beagle, and they have a very iconic woo-woo bark or howl. And the question is, do these breeds, although they're very different and probably have different selective histories, share signals in their genomes? And if so, then maybe those shared signals can narrow down for us what genetic elements affect vocalization. Well, just immediately looking at these population branch statistics across the genomes of huskies and beagles, I really don't anticipate there to be a lot of overlap. But nevertheless, I used Fisher's combined probability tests between windows across these breeds. Honestly, it didn't reveal any further clarity on the traits under selection. So regions differentiated strongly in one breed here really dominated, even if the other breed didn't have much differentiation at those loci. So this approach didn't really narrow down anything. And so it didn't really progress my goal of finding traits under selection. So I considered that maybe we could go more broadly and approach the grouping in a different way. What if we took the breeds that fall on either side of the spectrum for behavioral factors and tested whether their pooled signals are enriched for GWAS SNPs associated with the trait in question? And here's a mock-up of what I might expect if, say, the group of breeds that are highly biddable also had enrichment for biddability-associated SNPs among those genomic regions that are differentiated in those breeds. On the x-axis, I'll be plotting subsets of GWAS SNPs below a certain p-value threshold, increasingly more significant. And on the y-axis, I'll be plotting the proportion of PBS windows from the pooled set that overlap with those SNPs. At least at first, for physical trait differences, I really did see that this approach might work. We group dogs by either if they are just significant in general, significant in one direction, significant in another direction, or not significant at all. And what we see for height, at least, is that the set of small breeds have differentiation signals enriched in size-associated SNPs. We see some patterns of enrichment for other physical trait loci. The thing with a physical trait GWAS is that a lot of these are few loci with high effects. So that probably explains the drop-off here, is that there's really only one locus responsible for fur length in dogs. Oops, sorry. But when we apply this to behavior, I'm not really seeing enrichment here. It doesn't seem like this is providing any further clarity either on whether these behaviors that differ between breeds have some explanation in the genetic differentiation. So I thought, okay, well, for a lot of the GWAS, we have a manageable set of regions associated with traits, and we aren't necessarily interested in all of the SNPs. For instance, we have some pretty good associations for howling frequency, including 2 genome-wide significant peaks. So maybe there's a locus here that's differentiated in a particular breed or set of breeds. Perhaps breed ancestry in our diverse dog set is what's driving some of these associations. Or maybe it could be that howling in beagles and huskies is controlled by different genetic loci. Might we find that this locus is the Siberian husky locus and this is the beagle howling locus? So the way I tested whether trait-associated loci, like those in the howling GWAS, are more differentiated in breeds, is I randomly permuted the associated locus over 100,000 times. And then I mapped the maximum PBS from each breed to both the associated locus and its permuted loci. Then I scored the percentile that the real locus falls in relative to the null distribution formed by the permutations. And lo and behold, neither of the most significantly associated loci in the GWAS for howling were highly differentiated in our 2 iconic howling breeds. However, either breed did have significant differentiation at 2 different, less associated loci. So this seemed to be an approach that actually was working for what I wanted to find. And now if we take an overview of breed differentiation at all the height-associated loci, we see that the smaller sized breeds, like toy poodles, dachshunds, chihuahuas, all have more differentiation at more size genes. And nearly all breeds are also super differentiated at least a few size loci as well. So there seems like there's some sort of physical trait selection going on here. And indeed, in a very broad overview, we see a pattern that suggests breeds are under primarily aesthetic selection. Plenty of breed differentiation exists in loci that are associated with physical trait differences like size, spotting, tail curls, ear shape. But very minimal differentiation is found in loci associated with our behavioral factors. So there seems to be this primary aesthetic selection going on in breeds that will make it very difficult, especially in interbreed approaches to association, to find genomic regions responsible for behavior that differs between breeds. So although we know there are behaviors that differ between breeds, what kind of value can breeds at a genetic level serve us for finding what's responsible for those differences? And this is where I'm going to give a perspective on how breed ancestry and admixture might help. What have been principally neglected in most canine genomic studies are mutts, dogs that have a mixture of breeds in their ancestry. Darwin's Ark was created in part to address and include dogs of diverse ancestry in genomics research. And dogs like these might not only help us discern the role of breed ancestry and behavior, but also, owing to admixture in their genome, they could help us better map and segregate genetic loci that contribute to differences in phenotype. And because many pet dogs in the world, and most of the dogs in the world, don't come from pure breed backgrounds, to study variation in the dog population, we can't really ignore mutts either. We must, of course, infer ancestry from breeds in these dogs. So there are two measures of ancestry that we can consider here. First is the global ancestry, or the percent ancestry from those breeds. And then there's the local ancestry, the segments of the genome that were likely inherited from those populations. And local ancestry can be very difficult to estimate, given its reliance on proper phasing of the maternal and paternal chromosomes, the sharing of haplotypes between breed populations, and in dogs, the special problem that the number of populations we're looking for here can exceed 100. So local ancestry is something that we still haven't been able to infer very well in dogs. We use the software package admixture to infer global ancestry in the dogs of our genetic data set against a reference panel of 101 breeds. Some breeds are, of course, overrepresented in our data set, and that's because they're especially popular in pet dogs. Things like American Pit Bull Terriers are very popular, Labrador Retrievers, Chihuahuas. So we see that in the Darwin's Ark set. Sometimes dogs also carried ancestry from populations that aren't represented in our reference panel, resulting in what we call rainbow dogs. The dog on the far right, we know actually to be a street dog from St. Mauritius Island. So it's unlikely that any of the breeds detected in their global ancestry are actually breeds they carry. But so if breed behavior differences do exist and they have some genetic basis, then we might expect that there will be a relationship of ancestry with behavior in admixed dogs. So say the locus responsible for biddability, the high biddability we see in border collies, is carried here in the border collie genomes. At least in the F1s, there should still be a strong effect of ancestry from that one copy of the chromosome. But as recombination occurs, the haplotype carrying that effect might be kept and retained, or it might get lost. So that probability of a behavior affecting locus being retained in the local ancestry of admixed dogs should to some degree be reflected in the effects of global ancestry. Therefore, we considered constructing linear mixed effects models for behavior scores in admixed dogs to see whether the fixed effects of breed ancestry impact behavior. And we really only found two factors in a small set of breeds for which this is the case. So the strongest one by far is for biddability, the effect of border collie and Australian cattle dog on biddability, and conversely, the effect of beagle ancestry and Chihuahua ancestry on independence we found to be significant. This is a very distilled down breed effect from what we saw in the survey data, but it's still worth exploring. So going forward, looking ahead, the real key to using breed as a means to understand these breed-enriched behaviors is probably to get at measures of local ancestry and leverage that ancestry to improve our association mapping. So I'm looking to two approaches. RF mix here, unlike other ancestry inference methods, performs very well on more than 3 populations. It can even handle 100, corrects for phasing errors, and can even learn from the admix samples to boost performance. So it looks like a good choice for inferring ancestry in dogs. The next, from the Neil Lab and Dr. Atkinson, comes Tractor, a software package that considers local ancestry and association studies. And we might be able to use this to consider, say, border collie ancestry segments in associations for bidability. So with that, I want to thank folks from the Carlson Lab, both at the University of Massachusetts and the Broad Institute. And if you're interested in dog research and you have a dog at home, feel free to enroll them in our project, and they can help provide more insights into the basis of behavior in genetics. And I'll take any questions that people might have.
Thanks so much for your talk, Kathleen. That was really amazing. I want to remind the audience that if you do have questions, it would be wonderful to please post them in the Q&A, and I will voice them as soon as I can. So we'll maybe give people a minute or two to start typing things, Kathleen. So.
Okay. Thank you.
I guess in the meanwhile, as we're awaiting any questions that may arise, I want to ask, I thought this was really interesting. I believe it was about howling, how it seemed that there may be, many different routes to howling. I wonder if, how generalizable you feel that may be. That is, would your prediction based on that be that many of the traits that we might assume to have a shared genetic basis are actually look more like
differentiated in some way? Yeah. I mean, you can pretty much hear it in the Siberian Huskies and the Beagles that those are different vocalization patterns. But when I look at the genome-wide association across pet dogs for howling frequency, it's going to be a mix of dogs, Siberian Huskies, Beagles, other dogs that howl frequently. So yeah, those could be distinct loci, and I like this approach to applying the differentiation signals to fuss out which loci are enriched in which breeds. So
yes. Yeah.
That I'm going to apply that method to our other genome-wide association studies and see if I can piece out where the breed effects live. And maybe the variation in that trait is actually just additive small effects across the pet dog population, something not having to do with these breeds at all.
Yeah. Thank you. Well, howling turns out to have been a very popular topic because a question just posted by Shirley addresses howling as well, and I will just read that now. So first of all, she says, "Great talk, Kathleen." And then Shirley says, "For the howling trait, have you considered making it binary, like sometimes howl and never howl? And since the frequency of howling also depends on the environment."
That's a good question. I think that if I made it into a never howls versus howls at all, it might actually be asking about a different trait, which is there are some dogs that are just quiet and don't howl at all. So I think with the howling frequency, you're getting at a more frequent howling behavior, whereas if you make it binary, you're really asking about the inverse, doesn't howl at all. I want to try that. I think that a lot of the smaller breeds might not howl as much, so I wonder if we might come up with size associations too. Yeah, good question.
Great. I don't see any other questions at the moment. Let me, oh, actually, in the nick of time, another question. So this one is from Bob Kamsaker, and the question is, recently, a VNTR in the ACAN gene was shown to have a strong association with human height, up to 3 centimeters between the extremes. Mm-hmm. Do you have enough data on, do you have data on enough dogs from which we could evaluate whether ACAN varies in dogs and whether this could have an effect on height size?
Let's see. So there's actually multiple loci that go into affecting size in dogs here. Shirley actually from my group came up with a good predictive score of height in dogs, and she could probably evaluate the effect of that on predicting height in dogs too. Yeah, I'd have to dig into it, but it seems like multiple times in different species, height affecting loci have risen in selection, or it seems like there is kind of this whole group of or swath of different genes that affect height in different ways. So fussing out what the effect and what direction it goes, we could possibly do that in dogs, but we'd have to deal with these very high effect loci like the FGF4 retrogene, which is responsible for corgi legs. That's a big effect. So you have to consider that in dogs, we have some really big effect genetic variants here. So I'm not sure if any one particular gene we're going to be able to determine what the degree of effect is when you're in the context of these ones that determine your size from a Great Dane to a Chihuahua. Versus in humans, the height differences are more minute, more gradual, unless you're dealing with gigantism or dwarfism.